Hi students, this video will explain about the alternative mechanism of carbon fixation. The learning outcome is for students to be able to explain photorespiration and state the alternative mechanism of carbon fixation, which involve the C4 and crassulacean acid metabolism (CAM) pathways. Then, students should be able to compare carbon fixation in C4 and CAM plants. First, we need to understand why this adaptation occurs. Because most of plants, the C3 plants, have problem when they came across the hot and dry day or dry climate. Because during hot and dry day, photorespiration occurs. Okay, photorespiration. Photorespiration occurs due to the plants want to prevent from being dehydrated. If photorespiration occurs instead of photosynthesis, the potential or the yield of the photosynthesis will be reduced to 30 to 40%. Why can photorespiration occur? Because when the day is hot and dry, the stoma will close. Stoma will have a tendency to close to conserve water. So since the stoma is closed, it will prevent the diffusion of carbon dioxide from atmosphere into the cell. This causes carbon dioxide level to decrease. Why decrease? Because carbon dioxide that is available in the plants, in the chloroplasts, will be used during Calvin cycle. Carbon dioxide is used in Calvin cycle, but no additional carbon dioxide can diffuse from atmosphere into the cell through the stoma, because stoma is closed during the hot and dry day. So no increase, no addition of carbon dioxide concentration. That's why carbon dioxide level decrease. So because of this decrease, the low concentration of carbon dioxide, Rubisco, the enzyme that is supposed to bind the carbon dioxide to the RUBP, will have problem to bind carbon dioxide because Rubisco has higher affinity towards oxygen. Okay, once stoma close, carbon dioxide level decrease, oxygen gas increase. So because of this, Rubisco will have the tendency to bind oxygen to RUBP instead of binding the carbon dioxide. So this is why we consider oxygen as the competitive inhibitor. So because of that, RUBP will combine with oxygen, forming a 5-carbon compound instead of 6-carbon intermediate compound. The intermediate sepatutnya 1 tambah 5, 6. But the carbon dioxide cannot be bound to the RUBP. Instead, the oxygen will be bound to RUBP. So the products or the molecule formed from this binding is 5-carbon compound. Okay. So the problem during hot and dry day, stoma is closed to conserve water, carbon dioxide decrease, oxygen gas increase. Photorespiration occur. Photorespiration is when the oxygen is bound to the RUBP. So that suppose a normal carbon fixation when the carbon dioxide concentration is high, it should bind carbon dioxide to the RUBP by Rubisco. But because of the low concentration of carbon dioxide, Rubisco has high affinity to bind oxygen to the RUBP. Okay, not Rubisco bind with oxygen. Rubisco has high affinity to bind oxygen to RUBP, forming a 5-carbon compound. So after that, the 5-carbon compound will be broken into 2 types of molecule. First one is the phosphoglycolate, and the second one is the 3-phosphoglycerate, which is supposed to be 2 molecules if the normal condition is achieved, meaning a high carbon dioxide. So since carbon dioxide cannot be bound, instead the oxygen is bound to the RUBP. So instead of having 2 molecules of 3-phosphoglycerate, we can only have 1 molecule of 3-phosphoglycerate plus 2-phosphoglycerate. So this shows a reduce of photosynthetic output. Later on, we want to use the G3P, so we have to run more Kelvin cycle. We have to run more Kelvin cycle, or we have to produce more 3-carbon compound, the phosphoglycerate, to produce the G3P. So the yield of the output for Kelvin cycle will be reduced. So we cannot use this directly to produce the G3P. We can only use this one, so the output will be reduced. The phosphoglycolate will undergo a series of reactions split by peroxisomes and mitochondria that can produce carbon dioxide and use the oxygen and ATP. Then the carbon dioxide can be used in the Kelvin cycle. So photorespiration is considered as a wasteful process because we need to convert the phosphoglycolate to carbon dioxide first. Then we can get the 3-phosphoglycerate. So it takes time, longer and more steps to achieve the output. So photorespiration reduces the efficiency of photosynthesis to 30 to 40%. Scientists have discovered an alternative mechanism of carbon fixation to prevent the photorespiration. And these alternative mechanisms occur in C4 plants and CAM plants. So there will be a C3 plant that will continue with the reduce of output. Then we have the C4 plant, which is more efficient, overcoming the hot and dry climates, and also the CAM plant. The example of C4 plants are corn and sugarcane, jagung dan tebu, while CAM plants we have cactus and pineapple as the example. These alternative mechanisms can minimize photorespiration and then optimize Kelvin cycle even in hot climates, even when their stoma are closed. So the 2 alternate modes of carbon fixation are called H-slack or C4 pathway, H-slack or C4 pathway, which occurs in C4 plants. We name the plants that use H-slack pathway or C4 pathway as C4 plants, while for plants that have the crassulacean acid metabolism pathway is called CAM plants, cacti and pineapple. So both pathways actually precede the Kelvin cycle to produce sugar, meaning Kelvin cycle still occurs in these plants, in C4 and CAM plants. But before Kelvin cycle starts, they have the ability to bind the carbon dioxide even at low concentration. So that is why it is called C4 pathway or CAM pathway. Let's look at C4 plants first. The first adaptation of C4 plants is having a slight different anatomy compared to C3 plants. C4 plants have Crohn's anatomy. How do they differ? How does Crohn's anatomy different from C3? Okay, they have the mesophyll cells, same with C3, but the mesophyll cells in C4 plants are arranged in a ring form around the bundle sheath cell. Okay, C3 plants also have bundle sheath cell, but in C3 plant, the mesophyll cells are arranged loosely in C3 plants. In C4 plants, the mesophyll cells are arranged in a ring form around the bundle sheath cell. So this is the bundle sheath cell. This is the bundle sheath cell. And there are mesophyll cells that packedly arranged around the bundle sheath cell. Mesophyll cells and bundle sheath cells, they differ in a few parts of their structure. The mesophyll cells and bundle sheath cells both have chloroplasts, but the chloroplasts in the mesophyll cells are smaller and less compared to bundle sheath cells chloroplasts. The chloroplasts of mesophyll cells have larger grana and larger and more. The number of the grana is more and larger in size, while bundle sheath cell has less grana and smaller in size. And in mesophyll cells, there are few starch granules, tak banyak berbanding dengan bundle sheath cell. They have more starch granules. So, if we look at this diagram, this is the bundle sheath cell. This is the mesophyll cells arranged loosely and they do not surround the bundle sheath cell. While for Crohn's anatomy, the C4 plants, the bundle sheath cell is here and the mesophyll cells are arranged tightly surrounding the bundle sheath cell. Hatch-like pathway occurs in C4 plants. It is known as C4 pathway because the first product, the first molecule formed. The first molecule formed during the pathway is a 4-carbon molecule, which is oxaloacetate. While in C4 plants, both C4 pathway and C3 pathway, the Calvin cycle, occur in different cells. The C4 pathway occurs in mesophyll cell, while the C3 pathway occurs in the bundle sheath cell. So the mesophyll cell is the cells that surrounding the bundle sheath cell. So first, C4 pathway occurs in the mesophyll cell, and then it occurs in the bundle sheath cell. Hatch-like pathway shows there are twice carbon fixation. First is the C4 and the second one is the Calvin cycle fixation. The first carbon fixation is during the C4 pathway that occurs in the mesophyll cell. Okay, the first carbon fixation is catalyzed by Pep carboxylase, while the second carbon fixation is the C3 pathway, the Calvin cycle, that occurs in the stroma of chloroplast of bundle sheath cell. Bundle sheath cell. And as you know, the enzyme that catalyzes the carbon fixation in C3 pathway is rubisco. So this is the mesophyll cell where the first carbon fixation occurs. The enzyme involved is the Pep carboxylase. And then second carbon fixation, the Calvin cycle occurs in the bundle sheath cell. The enzyme is RUBP carboxylase oxidase. So what happened during the first carbon fixation? Carbon dioxide combined with the phosphoenol pyruvate or the PEP to form oxaloacetate. PEP is 3 carbon molecule combined with carbon dioxide. So it formed 4 carbon molecule named oxaloacetate. This reaction is catalyzed by Pep carboxylase. Pep carboxylase has high affinity for carbon dioxide. Higher affinity compared to Rubisco, meaning that it can bind carbon dioxide to the PEP even at low concentration of carbon dioxide. The truth is, Pep carboxylase has no affinity for oxygen, so oxygen cannot be the competitive inhibitor for Pep carboxylase. Then only carbon dioxide can be bound to the PEP. So the enzyme do not tolerate any other competitive inhibitor. Then oxaloacetate is reduced to malate. Okay. Reduced to malate means it uses NADPH. So NADP plus will be returning to the light dependent reaction. Okay, what happened to the malate? Malate will then exported from the mesophyll cell to chloroplast of bundle sheath cell. It will pass through the plasmodesmata. This is the plasmodesmata, the pore that can be found on the cell wall. So malate carried or transported to the bundle sheath cell, and then it will be converted or undergo decarboxylation to form pyruvate and carbon dioxide. See, malate will be converted to pyruvate and carbon dioxide. So the carbon dioxide will enter or will be bound to RUBP. Why? This is a step of accumulation of carbon dioxide. So the concentration of carbon dioxide in the bundle sheath cell increase because of this process. Then Rubisco will have the tendency to bind carbon dioxide to RUBP. Hence the Calvin cycle. So this is the Calvin cycle. Carbon dioxide then combined with RUBP, catalyzed by Rubisco and forming organic compounds, as what we have learned in Calvin cycle. Then what happened to pyruvate? Pyruvate will be transported back into the mesophyll cells and then undergo phosphorylation by ATP to produce to form PEP, phosphoenol pyruvate. Because of the C4 pathways, C4 plants can minimize photorespiration because the enzyme is more efficient. It has no affinity to oxygen, so it can bind carbon dioxide to PEP even at low concentration of carbon dioxide. So this is an advantageous adaptation in hot and dry day where stomach partially closed during the day. Let's look at the comparison between C3 and C4 plants. Okay, both require Calvin cycle to produce sugar. Calvin cycle occurs in both plants. But for differences, carbon in C3 plants Carbon dioxide fixation occur only once, while in C4 plant, carbon dioxide fixation occur twice. The fixation of carbon dioxide in C4 plant occurs in mesophyll cells and bundle sheath cells, but for C3 plant, it occurs in the mesophyll cell only. The carbon dioxide acceptor is RUBP for C3 plants, but for C4 plants, the carbon dioxide acceptor is PEP and RUBP. The enzyme also different, even though they have the same one. C3 plants only have Rubisco, while C4 plants have Pep carboxylase and Rubisco to catalyze the carbon dioxide fixation. We can say the first molecule that is produced in C3 plants during a light-independent reaction is 3-phosphoglycerate, while C4 plants, the first molecule formed is oxaloacetate. C3 plants considered less efficient, has less efficiency of photosynthesis, meaning the photosynthetic output is low during hot and dry day, while C4 plants have high efficiency of photosynthesis. The output is higher compared to C3 plants. Photorespiration occurs in C3 plants, but there is no photorespiration in C4 plants because Pep carboxylase will not bind oxygen to the PEP. C3 plants have no anatomy, Kranz anatomy, but C4 plants have Kranz anatomy. You can explain what is the meaning of Kranz anatomy. Let's look at crassulacean acid metabolism or CAM pathway. This pathway involves CAM plant, the crassulacean acid metabolism plants. These plants have leaf anatomy like the C3 plants, so they don't have the Kranz anatomy. In CAM plants, both CAM pathway and C3 pathway occurs at the same cell, which is the mesophyll cell, but occurs at different time. Okay, the carbon fixation occurs in different time. Okay, what happened? CAM plant during the day, the stoma are partially closed. This is to prevent dehydration, conserve water. But because of the partially closed stoma, the diffusion of carbon dioxide is prevented. But then they open the stoma at night. So the concentration of carbon dioxide is high at night. The pathway is similar, only it occurs at different times, similar to C4 plants. Look at this. Pep carboxylase catalyzes the binding of carbon dioxide to Pep. The same efficiency. So it forms oxaloacetate. Then oxaloacetate undergo reduction and produce malate. Okay, the difference is malate will be stored in vacuole. At night, it will be stored in the vacuole, and when the day comes, the malate will be transported out of the vacuole, and then will be converted to carbon dioxide and pyruvate. So the same thing actually occurs. Malate converted to pyruvate and carbon dioxide will enter Calvin cycle in the chloroplast. So it occurs in the same cell. This is the same cell. This is the same cell, only time different. So at night, Pep carboxylase are more active to bind the carbon dioxide with the Pep. Because during this time, at night, the carbon dioxide concentration increase due to the opening of stoma. But then the malate formed from this reaction is stored in the vacuole. When the day comes, the malate will be transported out of the vacuole and converted to pyruvate and carbon dioxide. So carbon dioxide can enter Calvin cycle. Okay, this is what I have been explaining before. Malate is stored in vacuole of mesophyll cells, and then when the day comes, stoma is closed. The malate is transported to the chloroplast of the mesophyll cell and undergo decarboxylation to form pyruvate and carbon dioxide. Then carbon dioxide will combine with the RUBP catalyzed by Rubisco. So this is where the Calvin cycle occurs. Then what happened to pyruvate? Pyruvate is transported back to cytosol of the mesophyll cell to be converted to PEP by phosphorylation by ATP. Let's look at the similarity between C4 and CAM plants. Okay, they have both oxaloacetate as the first molecule that's formed during the pathway, which is the 4 carbon molecules. But still, we cannot call CAM plant as C4 because CAM plant does not have Kranz anatomy. Both plants require Calvin cycle to produce sugar, and carbon dioxide fixation occurs twice. So bila carbon dioxide fixation occurs twice ni, means that we can elaborate more on the enzyme. We have PEP carboxylase and Rubisco. So the difference is C4 plants carbon fixation occurs in two different kind of cells, the mesophyll cells and bundle sheath cells. While for CAM plants, the carbon fixation, even though it occurs twice, but it occurs in the mesophyll cell only. In C4 plants, carbon fixation occurs during the day, while in CAM plants, carbon fixation occurs during the day and at night. So ada dua masa yang berbeza. Okay, for C4, different location, mesophyll cell and bundle sheath cell. The time is similar, masa yang sama, waktu siang juga. Atau dia tak ada perbezaan siang atau malam. For CAM, occurs in the same cell but different time. So why malate is stored in the vacuole? Because this is to prevent an acidic environment in the cytosol, because the malate needs to wait for the daylight to be converted to pyruvate. Malate is an acidic substance. If it stays in the cytosol, it will increase the acidity. So malate stored in vacuole. Thank you.